Hello everyone, this is a continuation of the previous video on the different types of biosensors, and last time we looked at the first two of the four most popular biosensors, and these were calorimetric biosensors and fluorescence biosensors. In today's video, we'll look at the two other types of biosensors. These are surface plasmon resonance biosensors and antigen-antibody interaction biosensors. First, let's start off with surface plasmon resonance biosensors. These SPR biosensors are analytical devices that measure changes in refractive index near sensor surface. Now, they're commonly used to study molecular interactions such as binding events between proteins, DNAs, or even drug molecules. Now, let's study the working mechanism of these SPR biosensors by looking at a diagram. First, a light beam passes through a prism and it reflects off a thin gold film on a sensor chip. Now, gold is chosen because it facilitates the surface plasmon resonance, a phenomenon in which free electrons on the gold surface oscillate collectively when it's exposed to light at a specific angle. Now, on the gold surface, a dextran layer serves as a scaffold for attaching bioreceptors, which are then customized for specific interactions being studied. Now that we understand the working mechanism of the SPR biosensors, let's see how they actually work in a case of real-life application by looking at their involvement in cancer drug discovery. Now, in the development of a cancer drug targeting the PD-L1 pathway, a key checkpoint in immune response modulation, researchers immobilize the PD-L1 protein on the SPR sensor. Now, when a drug candidate binds to the PD-L1, the refractive index near the gold surface changes, which causes a shift in the SPR angle. Now, this shift in the angle provides details on data such as binding affinity, wherein we are measuring how tightly the drug binds to the PD-L1 protein, but B, also kinetics, wherein we evaluate how quickly the drug responses and associates and dissociates from the target. Now, such measurements are critical in optimizing drug efficacy and minimizing side effects. Now let's look at the second type of biosensors, antigen-antibody interaction biosensors. Now, what are these type of biosensors? Well, these biosensors detect highly specific interactions between antigens, these viral proteins, and antibodies, immune proteins, and their ability to quickly and accurately detect diseases make them invaluable in clinical diagnosis. Now let's refer to this diagram for a more step-by-step breakdown of the working mechanism. The biosensor is functionalized with antibodies immobilized on the surface, as you can see, and these antibodies are specifically designed to capture the antigen of interest. Now let's explore the working mechanism of these biosensors by looking at their application in the COVID-19 rapid test. In a COVID-19 rapid antigen test, the sensor is functionalized with antibodies that recognize the SARS-CoV-2 nucleocapsid protein. When a patient's nasal swab is applied, the viral antigens bind to the antibodies, which generates a signal. And this really again depends on the sensor type. Electrochemical sensors detect a change in the current or the voltage, whilst optical sensors measure alterations in light properties. This mechanism enabled the widespread testing during the pandemic, which provided results as in little as 15 minutes. Now, after the antigen binds, the signal passes through a transducer that converts the binding event into a more measurable output, as we have studied in the previous videos. The signal intensity directly correlates with the antigen concentration, which enables more quantitative analysis. And that brings us to the end of our video. Thank you for watching, and please stay tuned for further medical innovation updates.